Welcome back, AP Biology students. In class, we've been talking about photosynthesis, and we've been really focusing on the C3 plant. We just haven't called them C3 plants. They're the majority of the plants. Today, we are going to start talking about C4 plants. So this is another way of going through photosynthesis. Okay, in class, we've been talking about C3 plants. And C3 plants are named C3, 3-carbon, because their first stable intermediate, 3PG, is a 3-carbon compound. So there's a problem with 3-carbon plants in that when they try to compensate for hot, dry environments, they're going to close their stomata. Now, if you remember, the stomata are little like pores in the leaves of the plant, and they let water out, transpiration, and they bring CO2 in, which the plant needs to fix through the Calvin cycle. So they're going to close the stomata so that they don't lose water and wilt and die. The problem with that is they're not going to get CO2. So the CO2 levels will decrease. Meanwhile, the oxygen levels are going to increase because the light reactions are still taking place. Oxygen has a greater affinity for the enzyme Rubisco than does CO2. So if CO2 levels are down and O2 levels are up, it's more likely that O2 will bind to CO2 it's more likely that O2 will bind to Rubisco than CO2 will. So then oxygen is going to be used for respiration. So this is called photorespiration. So it's respiration in the presence of light. You're using the oxygen and you're releasing CO2. So not very efficient. So this is the evolutionary advantage of the C4 plant. So the C4 plant is named because its first intermediate is called oxaloacetate, which is a 4-carbon compound. So carbon 4. CO4 and CAM plants have evolved to efficiently conserve water while still fixing carbon and making organic compounds. Okay, so how is it that a plant will go through photorespiration? So first of all, let's remember the enzyme Rubisco. So both CO2 or oxygen can bind Rubisco. Either way, Rubisco will make a PGA or 3PG, depending on your nomenclature. So CO2 is more efficient. It makes 2 PGAs where O2 only makes 1 PGA. So if CO2 levels are low but O2 levels are high, they stand a greater chance of binding Rubisco. So if that happens, then the PGA will be shuttled into the mitochondria rather than into the stroma of the Calvin cycle, of the stroma of the chloroplast. So the PGA goes into the mitochondria, and it will use up ATP in order to break down and release CO2. So this process uses ATP, which is energy inefficient. So rather than making organic molecules that can be then later broken down and make lots of ATP molecules, you're going to only use up the ATP. So you're going to run out of energy. So in this way, photorespiration can drain away as much as 50% of the carbon that is fixed by the Calvin cycle on a hot, dry day. So here in northern Michigan, well, I'm in central Michigan, but the northern United States, our plants, our grass burns up pretty well in the summer, and our grass is C3 plants. And C4 plants, we have something called crabgrass. Crabgrass goes wild during the summer. So when our regular Kentucky bluegrass is burning up, our crabgrass is thriving. And that's because our Kentucky bluegrass is over here running out of energy, and our crabgrass is over here making all kinds of organic molecules. So what does the crabgrass, the C4 plant, have that our Kentucky bluegrass, C3 plant, doesn't have? It has a way of efficiently using CO2 or conserving CO2 so that it can be used instead of oxygen. So other examples, sugarcane, corn, a lot of your annual plants, they minimize the photorespiration. So they're not going to use the oxygen to make PGA. They're going to use the CO2 to make PGA. So this gives them an advantage when it's hot and dry and when you have intense light. So let's look at this graphic down here. So this is a cross-section of a leaf. This circle right here, these are your mesophyll cells. This is the mesophyll layer. This is where the chloroplasts are found. So this is where your light reactions are taking place. In a C4 plant, these are found in concentric rings around the bundle sheath. So over here you see what's going to happen. This is your mesophyll layer, this concentric circle on the outside. This is the bundle sheath, so this portion in here. So they're going to separate where the fixation takes place and where the sugar production takes place. So in the mesophyll layer, we're going to fix CO2 into a 4-carbon compound called oxaloacetate. That will then be shuttled into the bundle sheaths. In the bundle sheaths, that's where we're going to continue the Calvin cycle and make our organic compounds. So C4 plants have a unique leaf anatomy. They have spatial separation, so they're going to put the fixation in one area of the leaf, and they're going to put the organic molecule formation in another portion, right? So the mesophyll layers are where we're going to fix the CO2, and the bundle sheaths are where we're going to release the CO2, and that allows us to keep the levels of CO2 high so we can keep using CO2 rather than oxygen. So one major difference in C3 and C4 plants is that spatial separation of the fixation phase and the synthesis phase. Another difference between the C3 plant and the C4 plant is the enzyme Pepcarboxylase. So remember, Rubisco binds both oxygen and CO2. Pepcarboxylase will only bind CO2, not oxygen. So it prevents photorespiration. So when CO2 levels are low, they have a greater chance of binding because it's not going to bind O2. So CO2 will bind with Pepcarboxylase and form that first 4-carbon stable compound called oxaloacetate. That oxaloacetate will then be shuttled into the bundle sheath, and it will then go through the Calvin cycle in a fixed form. So that allows for a more efficient delivery of CO2 to Rubisco. So the stomata don't need to be open as much because they don't need to take in as much CO2. So they're able to close up more often in the sun and conserve water loss. So just to reiterate that spatial separation of the carbon fixation phase and the synthesis phase, I like this big graphic. Here's your section of the leaf. You can see the stoma here, the stomata. And this lighter green, this is our mesophyll layer, so that's where our chloroplast is. And then we have a darker green and maybe purple. This is the darker green is our bundle sheath. And then we've talked about our phloem and xylem, the vascular tissue of the plant. That would be the purplish color. So we're going to fix carbon dioxide out here in the mesophyll layer. Here you go. With our Pepcarboxylase, we're going to make oxaloacetate. A reduced form of oxaloacetate is called malate. Malate is actually what is shuttled into our Calvin cycle in the bundle sheaths. So this is a more recently evolved process. So evolutionary time, C3 plants would be older than C4 plants. Although C3 plants are the majority of our plants, which makes sense. If they evolved earlier, they would be in existence longer, so they have a greater chance of survival. So only 3% of your plants are C4 plants. And finally, 61% of those are grasses. Not that you need to know the numbers, it's just telling you the majority of C4 plants are grasses. So just some reiteration, C4 plants use Pepcarboxylase, C3 plants use Rubisco. Pepcarboxylase has a greater affinity for CO2 than does Rubisco, so CO2 is going to be more efficiently fixed, and it is going to be turned into the 4-carbon compound oxaloacetate. That is how it got its name, C4. And the C4 plant, the oxaloacetate will be reduced into malate, which is then shuttled into the bundle sheath cells, and that is where the synthesis phase will take place. You still have the enzyme Rubisco in the bundle sheath, but by separating the area, the mesophyll, where the light reactions is taking place and producing oxygen, and keeping the CO2 in a separate location in the bundle sheaths, you're able to keep the CO2 levels higher than the oxygen levels, so you're able to bind CO2 with Rubisco at that time. Okay, another type of plant that has evolved to deal with hot, dry conditions are the CAM plants. So you have the C4 plants and the CAM plants. Examples of CAM plants are succulent, so the water-containing plants like jades, cacti, pineapples, those are pretty common examples. So where the C4 plant separates the carbon fixation and synthesis of sugars by space, spatial partitioning, the C4 plant separates those two processes by time. Temporal refers to time, so it has a temporal separation. The CO2 fixation happens at night, and the synthesis phase happens during the day. So again, this is to more efficiently fix CO2 so as not to go through photorespiration. CO2 is going to be converted into your oxaloacetate, into your malate. It's converted into an acid and stored during the night, so malic acid. So during the day, the malic acid is broken down and CO2 is released to the Rubisco. So the 4-carbon compound will break back into a 3-carbon compound, releasing the CO2. CO2 binds Rubisco and does not have to compete with the oxygen to enter into the Calvin cycle. So when it's extremely dry, the stomata will stay closed even during the night. And the oxygen that's given off from the light reactions of photosynthesis will be used for respiration. The CO2 given off during respiration is going to be used for photosynthesis. So you still have a connection between the photosynthesis and the respiration, the photorespiration. So this allows us to better look at the processes that are happening in the CAM plant, or the Creslucian acid metabolism. That's hard for me to say. So here it is at night. The stomata are open on the left here. The stomata are open, taking in the CO2 through the stomata in the mesophyll, and it will be converted using the same enzyme as C4 plants. It's called Pepcarboxylase. Pepcarboxylase will fix CO2 into oxaloacetate, a 4-carbon compound, which is converted into malic acid, malate. So the same thing. So that will then, this is all in the mesophyll rather than separating it into the bundle sheaths. So then during the day, this malic acid will release the CO2, so this keeps the CO2 levels high, release the CO2 to bind with Rubisco, and then the rest of the story. Then the CO2 is converted into PGA, into PGAL, and then into organic compounds and recycled into RUBP. All right, that's a lot of phases. So here's your 3 different processes. The C3 plant is your typical photosynthesis. This is what you've always learned. You got CO2 entering into the Calvin cycle, binding RUBP, and making sugar. Next, you have the C4 plant, which separates the processes into different areas of the plant cells. So you have the mesophyll, which is going to be where carbon fixation takes place. And you have the bundle sheaths, which is where the Calvin cycle is taking place. The CAM plants, they will separate according to time. This is all in the mesophyll. Carbon fixation happens during the night, and then the Calvin cycle happens during the day. So it's pretty confusing to learn alternate pathways because we're used to hearing and seeing and studying processes in one direction, and now we're giving you two different directions to study. So C4 and CAM, just a way of eliminating the photorespiration if possible, keeping CO2 levels high. C4 separates by space and CAM separates by time. So I hope that's helpful.